Okay, so in this video, we're going to be introduced to speciation. We're going to look at both pre and post zygotic barriers and what that means. But first, let's define what a species is. So we're going to talk about the biological species concept. And in the biological species concept, that we define a species as when two organisms choose to have choose to mate, and then in the wild, so not like in a zoo or in a circus or something, but in the wild they choose to mate, and when they do, they have viable fertile offspring. And so the word fertile means that the offspring itself is able to have babies, and then viable is like strong, healthy, robust, like is going to survive. So when we talk about two groups, if they do not meet these criteria, so if they don't choose to mate in the wild, or their offspring are sterile, or they don't make viable offspring, we call these two populations reproductively isolated. And if you think about that, isolated meaning separated, and they're reproductively separated. So for example, here you have some happy face spiders, and they look different, but they can interbreed, so they would be considered the same species. So a species, if it's defined as its members interbreed and produce viable, fertile offspring, what would you call it when two groups do not breed together? That's right, reproductive isolation. So when we look at what reproductive isolation means, and in terms of what a species is, there's two parts. We have they choose to mate, and when they do, they have viable, fertile offspring. So what would prevent two groups or two individuals from mating and lead them to becoming reproductively isolated, meaning they don't mate together, and keep them as two separate species? That's one question. And then if two individuals do mate, but their offspring are not viable or fertile, would they be considered the same species, right? So when we look at this, this is where these words pre and post zygotic barriers come from. Things that would maybe prevent two individuals from mating are called pre-zygotic barriers because they don't make a zygote. So one example is geographic isolation. They may be on different continents, and therefore they don't ever get to choose to mate. And then you have post-zygotic barriers, and in post-zygotic barriers, that's if two individuals do mate, what are some reasons why their offspring may be sterile or not fertile? Same thing, or maybe not viable, or that maybe they don't have offspring at all. So those are going to be your post-zygotic barriers. And if you're not making viable, fertile offspring, then you can't have like a hybridizing of these two species. So when we look at pre-reproduction barriers, this is going to be an obstacle to mating or to fertilization if mating occurs. Now, in my class, you have 5 to write down. Geographic isolation, I don't consider a true pre-zygotic barrier. It may lead to other pre-zygotic barriers, but geographic isolation is one of them. We have habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation. We're going to talk about each of these with a few examples. And so here are our pre-zygotic barriers. And then after we finish those, we'll go into our post-zygotic barriers. So now, how do these barriers show up? Well, first, populations, they may become isolated. They don't have to, but they may become isolated, meaning you have a population and now they're separate. So if you think about like the penguins from Happy Feet, you can imagine like an ancestral penguin population, and then the population of penguins got separated into two groups. Now you have some penguins that sing heart songs to attract a mate, and you have other penguins that build like pebble nests to attract mates. Now when they come back in contact, they no longer choose to mate together. So while though their populations are isolated, two populations are isolated, whether it's geographically or reproductively, they evolve independently. So this is where those 5 fingers of evolution matters. So when you have like a population that becomes separated into 2 groups, in one group you have natural selection, genetic drift, mutations, sexual selection occurring, and in this separate group over here you have the natural selection, genetic drift, mutations, sexual selection occurring. Now, you notice I didn't say gene flow. There's no gene flow between these groups. If there was gene flow, that means that they would be still mating together, but we've removed gene flow, so they're isolated reproductively or geographically or both. And with that, as they evolve independently and they go through their own changes, when they come back into contact, and if they no longer choose to mate, then they're considered to be 2 separate species. So let's look at geographic isolation. You have a population, and then somehow they become geographically isolated. And then while they're separate, they have their own evolutionary pressures and changes happening in these 2 separate groups, causing them to evolve to become different. And if when they are reintroduced together, if they still mate, then speciation did not occur. However, if they do come back in contact and they no longer recognize each other as the same species and they don't mate together, well, now they're 2 separate species. So we can see this with the Galapagos finches. So about 500,000 years ago, you have some finches from mainland South America land on an island. So this would be like the founder effect. They fall in love, they have some offspring, and then as the offspring and as the birds move to different islands, you have variation in the offspring first off, but each of the islands has different selective pressures, different foods, different plants, different resources. So you have natural selection occurring on each of the islands to favor different traits. So you have small finches, medium finches, large finches. You have finches that use tools. You have finches that eat insects, ones that feed on cactus flowers. So here now, today, we have 13 different species of finches on the Galapagos Islands, and yet they all evolved from one common ancestor. But once they got separated onto the different islands, you have them each evolving separate from the other finch populations into their own species. So that's geographic isolation. Then you have habitat isolation. So they may occur in the same region, but they occupy different habitats, so they rarely come in contact. So like the lion and the tiger live on the same continent, but they live in different habitats. So while they could produce offspring together possibly, as we'll see later with ligers, they may not actually do that in the wild. Now, you can also think about here we have some bullfrogs. You have, let's see, the northern red-legged frogs is isolated from the population of American bullfrogs. One lives in fast-moving streams while one lives in permanent ponds. So while they may live in the same forest, they're not going to ever, they're gametes especially, but they're not ever going to come in contact together. Sometimes in really large lakes, you can also see this where if you have like fish living near the top of the lake that feed on like flies or insects landing on the surface versus fish that live at the benthic zone or the bottom of the lake in the murky like depths, those 2 fish species aren't going to mate together. They live in different regions of that habitat. Now we also have behavioral isolation, which is one of my favorites. So with behavioral isolation, this is where you have unique behaviors or rituals to attract your mates. So this is when birds dance or sing songs. We saw the widow bird case study in class, but this is where they have different behaviors or courtship rituals or mating calls to attract their mates. So the dance that the blue-footed boobies do would only be attracted to other blue-footed boobies. And so, or on some species of frogs, their mating calls that they make, actually, their own species sometimes is the only species who can actually hear that mating call. Or when crickets chirp or birds sing, etc., those are all behaviors to attract mates of their own species. Then we have temporal isolation. And in temporal isolation, this is when species breed at different times. So basically different times of day, different seasons, different years. And if they're breeding at different times, how can their gametes possibly fertilize? right? So here you have American toads and Fowler toads. One is in early summer reproduction, and then one reproduces in late summer. How would their gametes ever get to be in contact with each other? Here you have two species of skunks. You have the eastern spotted skunk and the western spotted skunk, and they overlap in range, like where you find them in nature. However, one mates in late winter and one mates in late summer. So how would they ever get to breed together, keeping them as two separate species? So here, these two skunks, they are not geographically isolated. However, they are reproductively isolated because they don't ever get to mix their gametes. They don't ever choose to mate together. So that's just a graph kind of showing that. Then we have gametic isolation. And with gametic isolation, this is where sperm of one species may not be able to fertilize the eggs of another species. This is important in ecosystems like underwater with like the sea urchins, where the red sea urchin and the purple sea urchin both release their gametes into the water, and then the water will like mix them, but the sperm will not enter the egg of the other species. Therefore, their gametes don't ever fertilize, keeping them as two separate species. They never have viable fertile offspring. This is also important in like pollination, when pollen is plant sperm carried in the wind, landing on all different kinds of plants, but it will only fertilize its own species. So two reasons for gametic isolation. You could have the biochemical barrier or a chemical incompatibility. And so, but one reason or another, the gametes do not form a zygote. And then we have mechanical isolation. And in mechanical isolation, this is where the male and female sex organs do not really allow for sperm transfer. They don't fit together. And so this also applies to insects and other pollinators with the flowers. So if you look at like this hummingbird has co-evolved with these trumpet flowers. And so like the beak of the bird can fit into that flower for the nectar, but a honeybee would not be able to pollinate or transfer nectar from the trumpet flower because it's like a heavier insect that needs to land. So you can see on the foxglove purple flower how that honeybee can just like land on the flower, get the nectar, pick up the pollen. So bees would never pollinate trumpet flowers and vice versa. Okay, so now let's spend a few minutes. There's only 3 post-zygotic barriers. So now in post-zygotic barriers, this is where maybe mating mating occurred. But what are some reasons why you don't have a blending of these two populations to be one species? So this is, we're looking at 3 reasons why a hybrid offspring may not be viable or a fertile adult. So the first one is reduced hybrid viability. So the hybrid is the offspring, but it's not viable, meaning it's not going to survive. So if I would like to become a farmer one day, or like have land, and let's pretend I get some sheep and goats. They don't really have to pretend I'm going to get goats. So let's say I have some goats and then I get a sheep and they mate. That's likely to happen on the farm, but in utero, even if fertilization happens, during development, that fetus, it doesn't develop properly and the hybrids die. So therefore, you're never going to have a sheep-goat hybrid born. Same thing is happening here with these salamanders. Hybrids may form, but they do not complete development. And if they do, they're super frail and won't survive to adulthood. So remember, the hybrid is not viable. Like it may form, but it's not going to develop correctly. So here you have like a weak or not viable hybrid. Now, reduced hybrid fertility. In this one, the hybrids may be viable, but they're sterile. So this is a perfect example of a horse and a donkey, where horses have 64 chromosomes, but a donkey has 62. Their offspring of a mule is strong and a good work animal, but it's sterile. So you'll never have horses and donkeys blending to all become mules. And then our last one is hybrid breakdown. So maybe that first generation, the F1, is good and viable. But when an F1 and an F1 mate to produce an F2, now is when they break down. You cannot keep blending hybrids together probably from missing important genes and just not the correct proteins leading development in those F2 generation. All right, good job.